Benzopyrene is one of the main contaminants in air and basically in the environment. Also, this compound is known to have originated from incomplete combustion of organic compound. Chemical structure in general composed of fused benzene. Actually, in real life, we would come across this chemical in car exhaust, especially in smoke from tobacco, wood fire, or some other smoke of gas products, or it can be in water, and most likely we might also see it in grill food. As an analyte of interest to study because it's important to know how the dangerous the chemical is and how it works with HPL instrument. The abundance of this chemical in environment is a threat to air quality, and also many studies have related this chemical toxicity to human issue like skin tumor or long-term cancer and so on. When it comes to HPLC, HPLC work under the principle of thin layer chromatography, and indeed just like TLC, polarity play a role in the process. Two possible phases can be seen in this process, usually normal phase and reverse phase. Stationary phase is a solid like silica gel, while mobile phase can be liquid or gas. This manual is designed as a reservoir for the mobile phase. Usually the phase contains injected analyte, and in this case is benzene, benzopyrene and solvent. The pump is located right here and functions to push mobile phase through the system and to exit the column. Just one analyte is injected into the solvent machine. The sample is diluted and mixed with a mobile substance. In column, silica bits create a surface area for interaction to take place, usually between the component and the stationary phase. And lastly, after the component being separated and exit the column, in this area, a UV detector analyze the component and finally generate the signal. HPLC is a reliable tool for non-volatile compound. Column can be reused low sample consumption, also not limited to temperature stability. On the other hand, despite the process is costly and somehow sensitive to interference, this instrument still effective use to produce high resolution results. So in this lab, from this useful plot of retention time and intensity produced, we can easily calculate the concentration precisely and also to identify easily compared to other instruments. Data analysis. HPLC can be said to have a high reliability. In a study in which many samples have been tested, let's say like 40 samples, HPLC produce a high linear response at low tested concentration range. The reported R square is like 9.99, meaning that only 1% is error. Actually, on average, the recovery of benzopyrene from food tests also reported relatively high, which is about 81% with standard deviation 1.5%. In other words, the important thing is that the data here is pretty optimal to be reliable on the fact that most grilled food contain a lot of benzopyrene, and the numbers here exceed the maximum level of safety either by cooking with charcoal or with gas. Separation instruments like HPLC will affect many aspects of our life. Indeed, without this instrument, our world will be a less clean and also unhealthy place. The abundance of benzopyrene in food is still an important discovery, and actually HPLC And other similar instruments have made the way for later on science as well as to build a baseline for future experiment. And so building off the past, we would expect to see liquid chromatography to be more affordable and also improvement in order to deal with the existing constraint. And that's all for my presentation.